Hi, so for our cell signaling video project, we're going to be talking about the PDGF signaling pathway. The PDGF signaling pathway describes how cells use external signals to regulate functions such as growth, survival, and wound healing. PDGF is a growth factor that helps to bind receptor tyrosine kinase, or RTK, on the cell surface, triggering intercellular events. This signaling is extremely important in tissue repair, cell proliferation, which is basically increasing the amount of cells through growth and division, and embryo development. However, when not controlled, it can lead to diseases like fibrosis and cancer.
The signaling molecule PDGF is hydrophilic because it doesn't pass through the cell membrane and thus binds to receptors on the extracellular surface of the membrane. The receptor protein is a transmembrane protein, meaning it spans the entire cell membrane. The ligand binding site faces outside the cell, which is where the PDGF binds. So, what is this group of receptor proteins typically used for in the cell? PDGF binds to receptor tyrosine kinase, or RTK. The PDGF specific receptor protein kinase is responsible for controlling blood loss after a skin injury. It's also essential in wound healing and forming new blood vessels. There are three advantages of using receptor tyrosine kinase. The first advantage is amplification. This is where a signal PDGF binding could activate multiple signaling events. The second advantage is specificity. Different RTKs can activate different pathways. And the third advantage is flexibility. RTKs could also trigger many intracellular responses at the same time. However, there are also disadvantages of using receptor tyrosine kinase. The first disadvantage is that it requires energy. It requires proteins and ATP as energy in order for phosphorylation to occur. And it's also vulnerable to mutation events. If RTK is not regulated, it could lead to uncontrolled cell growth, which can ultimately lead to cancer. After a skin injury, PDGF binds to its receptor protein kinase at the site of the wound on the cell surface. This activates the receptor, and these two receptor proteins come together in a process called dimerization. Each receptor protein phosphorylates, meaning it adds a phosphate group. The phosphate groups serve as binding sites for intracellular proteins. Downstream cascades are activated, and the cell thus carries out changes in gene expression.
In the PDGF pathway, the stimulus is tissue damage, such as a wound, which causes platelets to release the signaling molecule PDGF. The PDGF binds to receptor tyrosine kinase, which is the receptor protein on the surface of the cell. Inside the cell membrane, the main molecules involved in intracellular response include adapter proteins RAS, cascades of kinase such as RAF, MEK, and ERK, and transcription factors, which are proteins that help to regulate gene expression. The sequence of events in the signaling pathway first include PDGF, which is released by platelets that bind to the RTK receptor on the cell surface. This binding causes the two receptor proteins to come together, or dimerize, as we talked before, which basically activates their kinase function. In the dimer, each RTK receptor phosphorylates tyrosine residues on the other intracellular tails, which basically adds phosphate groups and forms binding sites for downstream signaling molecules. These phosphorylated sites act like docking stations for intracellular proteins. For example, adapter proteins like GRB2 and SHC. GRB2 will recognize and attach to these tyrosines and then recruit another protein called SOS. SOS basically activates one major protein, RAS, which then triggers a kinase cascade, where RAF activates MEK, which activates ERK. Finally, the activated ERK enters the nucleus and activates transcription factors, which switch on genes that drive the cell cycle and promote cell growth and division. To keep the pathway from running forever, it must be terminated. Termination happens when the PDGF ligand degrades or is removed from the receptor. Phosphatase, which are enzymes that remove phosphate groups from the receptor and the signaling molecules, shut down their activity. RAS activity is shut down by RAS-GAP, which accelerates the hydrolysis of GTP. These steps overall prevent signal and cell growth.
An example of an antagonist of the PDGF signaling pathway is imatinib, which is a targeted cancer therapy drug originally developed to treat chronic myeloid leukemia, but it also blocks the signaling of PDGF receptors. It's an exogenous molecule, meaning that it comes from outside the body. Usually, when PDGF binds to its receptor, the receptor kinase dimerizes, then autophosphorylates, and then activates the RAS kinase cascade. However, imatinib blocks this by binding to the receptor's ATP binding site, thus inhibiting ATP production. Without ATP, the receptor cannot phosphorylate itself, and thus the signaling cascade shuts down. The pathway is blocked, and thus ERK cannot activate transcription factors in the nucleus, and as a result, the cell cannot turn on genes for cell growth and division. Imatinib prevents cell proliferation, and that is why it's an effective cancer therapy.
Here are the sources that we used, and we hope you learned a little bit more about the cellular pathway that goes into repairing your tissues after a wound. Thank you for listening.